Gametogenesis. Gametogenesis is the process of formation of gametes. The primary sex organs, the testes in the males, produce sperm, and ovaries in the females produce ovum. Gametogenesis is further classified into 1. Spermatogenesis. 2. Oogenesis. Spermatogenesis begins at puberty. Significant increase in the secretion of gonadotropin releasing hormone, which stimulates the pituitary glands and stimulates the secretion of 2 gonadotropins. Luteinizing hormone LH and follicle stimulating hormone FSH. LH acts on the Leydig cell and stimulates synthesis and secretion of androgen. Androgen in turn stimulates the process of spermatogenesis. FSH acts on the Sertoli cells and stimulates the process of spermatogenesis. Immature male germ cells, spermatogonia, produce sperms by spermatogenesis. Each spermatogonium is diploid, which divide by repeated mite division containing 46 chromosomes. Spermatogoniums present on the inner side of seminiferous tubule multiplied by mite division, forming primary spermatocyte. Primary spermatocyte undergo meiotic or reduction division, forming secondary spermatocyte. Secondary spermatocyte undergo second meiotic division to produce 4 equal haploid spermatids. 4 equal haploid spermatids transform into spermatozoa sperms by a process called spermiogenesis. Sperms move from seminiferous tubules by process of spermiation. The seminal plasma along with the sperms constitute the semen. The functional sex accessory ducts and glands are by testicular hormones, androgens. Structure of a sperm. It is a microscopic structure composed of a head, sperm head contains an elongated haploid nucleus. The anterior portion of which is covered by a cap-like structure called acrosome. The acrosome is filled with enzymes that help fertilization of ovum. The middle piece possesses numerous mitochondria which produce energy for the movement of tail that facilitates sperm motility, essential for fertilization. The human male ejaculates 200 to 300 million sperms during coitus. 60% must have normal shape and 40% show vigorous motility. Oogenesis. The process of oogenesis involves development of an ovum which starts from a primordial oocyte around which 2 to 3 layers of cells from the ovarian stroma. The outer layer is fibrous and known as theca externa. An inner layer, which is somewhat vascular and cellular, is theca interna. The cellular mass is soon separated into 2 parts by the collection of fluid, the liquor folliculi near its center. A homologous membrane, the zona pellucida, appears between the primary oocyte and the follicular cells. These cells increase rapidly, forming many layers. The theca layer are differentiated and the follicle assumes a deeper position in substance of the ovary. The primary oocyte divides meiotically, giving off the first polar body and becomes a secondary oocyte. The germ cells become mature ovum after the second polar body is released. During later stages, the fluid film spaces, the atrium appears in the membrane granulosa layer formed from the cells separated off by the liquor folliculae. The resulting structure becomes a vascular or graphene follicle. As the follicle matures or ripens, it projects out from the ovarian surface as a small cyst-like oval swelling which bursts to liberate the ovum, the process being called ovulation.
Your due date is calculated from the first day of your last menstrual period. Conception occurs about 2 weeks from this day, and that's when you're truly considered to be pregnant. During a menstrual cycle, one of your 2 ovaries matures an egg within a follicle. Hormonal surges controlled by your brain then cause 1 and occasionally 2 follicles to release an egg. If both ovaries release an egg, you may have non-identical twins, also known as fraternal twins. Long, finger-like projections sweep the egg into the fallopian tube. The egg travels down the fallopian tube, pushed by tiny hairs, and awaits the arrival of sperm. About a teaspoon's worth of semen enters the vagina, containing roughly 300 million sperm. Less than 100,000 will pass into the cervix to begin their 6-inch journey to the egg. A long, whip-like tail propels each sperm toward the waiting egg. Most sperm will lose their way. Only about 200 sperm successfully reach the egg. Fertilization occurs when 1 sperm penetrates the egg, combining their genetic material. The sperm and egg each contain 23 chromosomes holding all of the information that determines your child's sex and contributes to the child's genetic makeup, including physical appearance, intelligence, and personality. It's a boy or a girl. You just don't know which yet. Within 24 hours of fertilization, the egg, now called a zygote, divides into 2 cells. The number of cells doubles about every 12 hours as the zygote continues its trek to the uterus. During this early stage, if the cells split into 2 separate groups, instead of remaining attached, they'll be doubly blessed with identical twins. About 3 days after fertilization, the zygote has become a ball of 32 cells resembling a miniature raspberry. A few days later, it reaches the uterus as a ball of roughly 500 cells surrounding a fluid-filled cavity. On day 7, the ball of cells burrows into the uterine wall during a process called implantation. Many women have implantation bleeding or spotting at this time, which occurs about 21 days after, before a pregnancy test could even be positive. The cells on the outside of the wall create the yolk sac and the placenta, which nourishes your developing baby. By 38 weeks, the fertilized egg has grown into an infant with more than 2 trillion cells.
Development of the placenta begins during implantation. When the blastocyst adheres to the endometrial epithelium, trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytiotrophoblast layer. As the cytotrophoblast proliferates, newly formed cells migrate into the syncytiotrophoblast and lose their cell membranes, forming a rapidly growing multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytiotrophoblast sends out finger-like projections, allowing the blastocyst to embed into the endometrium. Lacunae, or spaces, begin to form within the syncytiotrophoblast. As the syncytiotrophoblast erodes the endometrial blood vessels and glands, the lacunae become filled with maternal blood and glandular secretions. Isolated lacunae fuse to form a network through which maternal blood flows, thus establishing early utero-placental circulation. Toward the end of the second week, small projections of the cytotrophoblast begin to expand into the syncytiotrophoblast, forming primary chorionic villi. Early in the third week, extra-embryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point the structures are known as secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the extra-embryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. Cytotrophoblast cells from the tertiary villi grow toward the decidua basalis and spread across it to form a cytotrophoblastic shell. Villi that are connected to the decidua basalis through the cytotrophoblastic shell are called anchored villi. Villi growing from the sides of the stem villi are called branch villi. These branch villi are surrounded by the intervillous space and will serve as the main sites of exchange between mother and fetus. By the fourth week, fetal blood flow is established. Poorly oxygenated blood is transported to the placenta from the fetus via paired umbilical arteries. These arteries send branches into the chorionic villi and divide into capillary networks at the terminal ends. Carbon dioxide and waste products are removed from the fetal circulation across the placental membrane to the maternal blood in the intervillous space. Oxygen and nutrients are transported across the placental membrane from the maternal blood to the fetal capillaries. Oxygenated fetal blood travels back through veins, converging to form a single umbilical vein that goes to the fetus. Blood returns to the maternal circulation through endometrial veins, forced out by the pressure of incoming arterial blood. The placenta nourishes the fetus throughout development and is delivered shortly after parturition.